From Microbe TV, this is TWiEVO, This Week in Evolution, episode 107, live streamed on Wednesday, October 30th, 2024. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Nels LD.
Good to be with you from LD Lab Studios. Almost snow-capped LD Lab Studios, it turns out, Vincent. We've started that transition, so if I'm looking out my window at the higher elevations, there's some evidence on the mountaintops that winter is on the way.
Cool. Are you a skier?
Yeah, so I grew up mostly cross-country skiing in the upper Midwest, but have diversified my portfolio since opening up LD Lab Studios and LD Lab here in Salt Lake City. It's so easy to get to multiple ski resorts and hop. You can be on, out of your office and onto a chairlift in less than an hour. So it's pretty spectacular. Yeah. Yep. How about you? Are you a skier out there in the Northeast? There's some great terrain.
I used to cross country when I started, but it got harder and harder to find good snow here in the Northeast. I mean, I remember taking a ski vacation one winter. There's no snow. It's just no snow. So I started going to those Keystone meetings, right?
Good idea.
Which are at ski places. So I said, why don't I learn how to ski? So I took lessons at the meetings. And, I was already in my 30s, kind of late to learn, but I got to be a reasonably good downhill skier. I could handle the blue slopes, but never the black diamonds very much. I wasn't very good at moguls, and I just got rid of my skis. Actually, I had a pair of Vocals, really old skis that. The modern skis turn for you now, right?
It is a sea change.
You had to do it yourself. You had to turn yourself. They were just flat boards. They were really good skis, but I just gave them away a few months ago, and I'm not going to ski anymore. Mainly, Nels, because people are too crazy on the slopes. They just dash. I'm afraid they're going to smash into me, okay? Because they have no control.
Yeah, that's fair. I've seen that happen, or some near misses for sure. But I ski on Vocals too, a little more newer vintage. And you're right; it's wild how good the equipment has become. I'm actually probably out of date right now as these things continue to improve.
I once went to a ski slope in Pennsylvania, and they had all these skis nailed to the wall like relics, and they had a pair of my Vocals nailed to the wall. Boom.
This might be a sign you're a little behind the times on.
So, sometimes I would rent those curved, those very fancy skis that basically turn themselves, right? You just give a little signal, but I never did, and now I'm out of the game. That's it.
Fair enough. You mentioned the Keystone meetings, and this is worth spending a moment on, which is this sort of magical idea, right, of a science meeting where you spend a significant amount of time, not just in front of the posters or all together with the PowerPoint presentations, but you're out on the ski slopes for, half a day and can really interact in ways that you are a little less formal. What a great way to do it outdoors. And so some good energy, some good ideas and innovations that have come out of some of those conversations.
Welcome, everybody.
Yeah.
Hit the like button if you haven't done so yet. It helps other people discover this. And thanks to our moderators for being here today. We have Andrew from New Zealand. We have Barb Mack, UK. We have Tom Steinberg from Oregon.
Great.
Thank you all for coming and moderating. We appreciate it. And all of you, tell us where you're from. We'll go through and check out your locales before we dive into the origins of life.
That's right. We're going
Eukaryotes anyway.
Eukaryotes. We're doing a deep dive. Not that deep. We'll settle for about 1 billion years instead of closer to 4 billion. But that's not a small distance in time there.
So Tom says he's hoping the forecast rain is sufficient to allow Chinook salmon passage to our Oregon Coast Range Creek to spawn.
Yeah.
Speaking of, this should have been my pick today, but I was watching some videos where they're knocking down dams in Oregon. right?
Yes.
So they built them in the early 20s, and
Hmm.
they screwed up the ecology so much
Yeah.
they realized the mistake and they're taking them down. But a dam is a big thing. And so you
Yeah.
must know all about this, Tom. And I watched
Yeah.
videos where it had one dam where they had built stairs for the salmon to go back, because the dam
Exactly.
screw up
Ladders.
the salmon's ability, but
Yep.
it just flooded everything. And so they got rid of them. You can't
No,
just release all the water. You have to be careful of
that's right. No, I agree. This is absolutely spectacular. I saw some of that video footage too. There's something, I mean, these are these salmon returning for like the first time in some cases, almost 100 years.
yeah.
And they're near the end of their, life cycle as they're preparing to spawn, and they're almost like disintegrating right before your eyes. But the fact that they're like how quickly some of this can be recovered by, taking these steps, it's really inspiring. A few years back, the Elwha Dam up on the Olympic Peninsula, a little bit north of where what you're describing, Tom. Of course, I'm sure you're completely familiar with the details here as well. But that landscape has just like as the sediment came down, and this is all the leading into the ocean there, the Harrow Strait, I believe. It's just absolutely gorgeous to see how that land kind of recovers
Mm.
or gets back to where it was after those dams were there for decades or even longer, centuries in some cases. Really excite, sort of beacon of hope. Seeing those fish come back and invest their last bit of energy in the next generation of salmon to come. Yeah.
Elizabeth is here from West Virginia. Nice to have you. Barb Mack is
Welcome.
from the UK.
Awesome.
There's Andrew from New Zealand.
Good to have you along as always, Andrew.
Pete is in a gloomy London.
Gloomy London. Yeah.
Abdul Rahman is from Albany, New York.
Welcome, welcome.
Paul. Hello, Paul. Welcome. Nice to see you.
Yeah.
Mark is in Helsinki. Oh,
Ooh,
5C and dark. Yeah, it's in the middle of the night there, right?
sauna weather.
Pete says, I need to get my old skis from the late 90s.
Now we're
Yeah.
talking. Yeah.
Nail them to the wall.
Relics.
Simon is from San Jose. How's
Mm.
Mpox, influenza, and SARS-CoV-2 evolving? I'm sure they're evolving just fine right now.
Yeah, they don't take vacations. They keep
Evolve
perking.
continuously.
Keep perking along for sure.
There's a new SARS-CoV-2 variant that's overtaking the others,
Yeah.
and this happens de rigueur.
Oh yeah.
Simon says the origin of life didn't it come from ancient aliens and them having an accidental lab leak? Yeah.
Yeah. You never know. We'll start with our minds open to all ideas, and then we'll maybe filter down a little bit based on some of the evidence that we find in front of us. But
Nels, if you find life on another planet,
yeah.
let's say on Mars where there seems to be some water or evidence of past water. I mean,
Hmm.
is it going to be similar to here, or I mean, it's so close, right? Relatively speaking.
Yeah.
But
I don't know.
what is it? An independent origin from Earth? It seems unlikely, doesn't it?
I don't know. I think again, anything's possible at some probability.
You
Does
don't
it
have all
use?
the answers, man.
I don't, but it does. I mean, actually, this is an important. This is like we're kind of sort of almost. This is a great intro or preamble to our discussion. There's a lot of overlap in thinking about our own deep evolution, but how that relates to as you really go back a billion years, there's enough time for a lot of things to happen. And so in cases of other alien origins of life, would it use DNA? What would be the sort of chemical basis of inheritance? Um,
Mm, yeah,
yeah, it's really fun to think about that. And I
because
think, really important to stretch the mind along these lines because, and we'll sort of put this into the framework of today's preprint in a few moments, the origins of eukaryotes. But yeah, you kind of have to, this is a field where imagination is important to sort of open up these possibilities and think really broadly. But that also, I think highlights some of the challenges of sort of transitioning from imagination to sort of scientific progress or to try to, diminish some of the unknowns. So yeah, this is great preview.
we're joined by moderator Vanity Nutrition. Thanks for joining us.
Awesome.
Vanity.
Welcome.
Yeah, it's just that Nels,
Yeah.
Mars being so close to Earth, you're almost tempted to think that, okay, this life
Right.
evolved on Earth, and then bits of it somehow got transported
Oh
elsewhere.
yeah, yeah, yeah. No, I think that's a really famous idea. Is it polyspermy or something like that, where
Mm.
either by asteroids or pieces like there's. And I agree. I mean, I think proximity is important. The things that are closer are, again, from a probability, more
Yeah.
likely to sort of communicate or exchange. And then at the same time, we've got all these visitors. I didn't. Did you see this comet that came by? This is a couple weeks ago. I think it was sort of right after everyone was putting up their sort of slightly enhanced or doctored iPhone photos of the northern lights, the aurora borealis, which made it here, at least with long exposures on iPhones. You can kind of see this. There's also a comet that came by, and so it was in the west sky here, and I just kind of randomly put my iPhone, not randomly, but sort of in a low probability gesture, put my iPhone above the sort of horizon line and got a photo of this thing that I couldn't see with my eye, but my iPhone got it. And so,
Mhm.
right,
That's why
did
they
it
call it an
like
iPhone.
did that? Events like this, either all of the shooting stars that are um, actually burning up in our atmosphere and the little particles and meteorites and
Yeah.
pieces that come down. That's right. That could be a source either of at the origins of life, or could be even involved in sort of hidden ways in the origins of eukaryotes that are, yeah.
Well, 2 weeks ago on TWiV, Kathy Spindler showed us. She
Hmm.
took a photo of the comet that was visible, and she couldn't see it, but she took a picture of the part of the sky where it was supposed to be, and the phone
Yeah.
showed it. Yeah, it's
Yeah. Same
amazing.
here. Exactly. Kathy and I converged on the same, same
Yeah.
thing there. That's awesome. I love it.
So it's panspermiology. That's the word.
Gotcha. Yeah. Yeah. Where the basically
Yeah.
life is seeded from meteorites, etc.,
And
shooting
yeah,
stars. So,
meteorites certainly get here from Mars and vice versa. Yeah.
yeah,
Ah,
exactly.
here's a good
Yep.
one. Abdur says, have there been 80 modifications on Miller-Urey? Oh, yeah. I think Miller-Urey is out, frankly. If you read any
Hmm.
of Jonathan, the guy in the UK, Jonathan, what's his name? He writes all the books on evolution, origins of life. I can't
Oh,
remember his name
yes.
now, but he said the Miller-Urey is just nonsense. What happened was life emerged down at the vents at the bottom of the ocean,
Yeah.
where you have this, I think it's alkaline seawater, and underneath is water with a different pH, and it sets up a gradient, which is basically like a
Mhm.
mitochondrial membrane, and it makes energy. Oh, it's beautiful. You have to read his books. What is
Yeah.
his
Yeah.
name? Jonathan King. King, the guy who does the evolution. No, that's not him.
We're not
Mm,
talking about Nick Barton, are we?
Nick something. Nick,
Nick Barton, maybe?
not Barton.
Okay. Yeah, we're sort of circling around it at the moment. If anyone out there,
Nick Lane. Nick
Nick
Lane.
Lane, yeah, there it is. Yep.
So,
Yep.
a terrific series of books, and
Hmm.
he will talk about Miller-Urey, which they taught us, in college. You put some chemicals in, and you put lightning, electricity, and it becomes organic matter.
Right. 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 Right.
And he basically says that's all. It can happen in the lab, but that's not how it happened.
Yeah, and so this is another example, of this field, which I think is fast. It's really hard because the events that the scientists are trying to capture, or our, fellow humans are trying to capture, are what happened billions of years ago, which our brains, at least my brain, can't even wrap its head around 100
Yeah,
years,
yeah.
let alone, all of the orders of magnitude more here. And so I've been kind of, one of the reasons. So actually, why don't we hop in here to the paper, and I think
Yep.
this will really nicely
Let's
dovetail with
bring
our
it up here.
conversation. So the preprint we're up against today is titled "Diverse Ancestries Reveal Complex Symbiotic Interactions During Eukaryogenesis." Um, and so, eukaryogenesis, origins of eukaryotes. So these are our cells now in a multicellular formation, but these are cells that have mitochondria, in contrast to bacteria and archaea, which don't. And so, what really went on, and there's been plenty of work in the field. Before we kind of set that up, let's highlight the authors here. So this is a group that we have three co-first authors coming out of senior author Tony Gabaladon's lab. And it's going to get worse when I try to pronounce the first authors, just warning everyone, and apologies to the authors. Is there, Vincent, is there like an AI chatbot that can pronounce names for you? I've got a knack
Good
for
question.
picking like great scientist names, which I then do a terrible job of pronouncing them. Anyway, so this is coming out of the Barcelona Institute of Science and Technology. And also, importantly, I would say the Barcelona Supercomputing Center. A lot of data crunching involved in today's preprint. This is up on the bioRxiv, freely available. Hasn't been peer reviewed. We're doing some podcast peer review and some live stream peer review with our audience today. And who else here? So Madrid, Spain, and then the Catalan Institution for Research and Advanced Studies. First author, Moises Bernabeu. We've got Sayo Manzo Morales and Marina Marseille Huben. Again, massive apologies for my pronunciation there. But this is this is Deep Evolution, the origins of eukaryotes, and then what are the contributions, the genetic contributions. The most obvious sort of glaring example we've already hinted at is the mitochondria, the so-called powerhouse of the cell, and of the sort of a hallmark of eukaryotic cells, a clear engulfment of a proposed proteobacteria, although we'll talk a little bit in more detail about that. Other contributions, genetic contributions, both horizontally and meaning that the genes are acquired somehow. And the thing that really kind of got my eye on this one, Vincent, was real contributions proposed from viruses and specific classes of viruses in that genetic conversation as sort of a vehicle to traffic genes is kind of, I think, given a standing that maybe has been not so much in this conversation up until now. And so, again, a field, I think, historically before the genome era that we're in now, or post-genome era, however you describe it, a field built on imagination and argument. So I can remember as I was starting to approach evolution as a kind of in the transition in my scientific training from grad school, which is a lot more kind of traditional cell biology, molecular biology, being at meetings and seeing old people yelling at each other about these, no, it wasn't the Miller-Urey, it was not, or the this or that. And in one sense, it was sort of, a little off-putting, just that the personalities were taking up kind of so much of the oxygen in the scientific conversations. But it was also, behind the scenes there, it was sort of, I think, the state of the art up until in the last 20, 30 years or so, as the genome databases have just swelled with more data. How you don't have time machines, and so you're kind of depending on imagination and then persuasion. All right, of you're like, well, here's an idea, and here's why I think this matters. And at least in some of the meetings I ended up sort of surfing into, it got wild in there. People were really, sort of emotional about their ideas and their creativity and how that was being critiqued and sort of compared as people were trying to advance these ideas. So I think it's kind of fascinating, both from a sociological standpoint as well as a scientific one. And maybe I'll return to that at the end. But there have been some important advances that I think are starting to maybe balance the field a little bit, or changing the conversation, bringing more data into the mix, right? And so, and I'd say there's sort of two categories here. So one is that scientists will discover new creatures, new critters, mostly microbes, and add these branches onto the tree of life. You and I have talked about this, Vincent. This is episode 91 of TWiEVO, not too long ago. Meet your very distant cousins. Do you remember that conversation a little bit? Yeah, that was really fun. So these are, there's sort of a cottage industry now, including scientists from the Netherlands who kind of get their courage up and send robots into really deep ocean territory off the coast of Scandinavia, get down to those thermal vents where some of the Miller-Urey related chemistry has been sort of floated, and find these modern species, mostly archaea, the Asgard archaea, that are just as modern as we are, but have kind of stayed in environments that are probably a lot closer to the kind of chemistry, the biochemistry that was happening as as eukaryotic cells originated or emerged on the scene.
Nels, what is an archaea?
Yeah, so archaea. Thank you. Yeah, let's keep positing as we're
I
gonna get
mean,
into this
I
is
know
the easy part.
eukaryotes. I know
We're gonna,
bacteria.
yeah, right. So archaea
I
is
know.
another branch of life. Sort of kind of an orthogonal take on bacteria in some way. Is that a fair? How would you describe it? So these are
Well,
microbial, right? Yep.
they have some characteristics of eukaryotes, right? I mean,
Oh yeah, yeah,
morphologically, they look bacteria. Rather
yeah.
simple cells. They don't
That's
have a
right.
nuclear membrane, I believe, right?
Yep.
Um, but they are different from bacteria. They have some features like lipid compositions that resemble eukaryotes as well, and they hadn't been discovered until not too long ago, right?
That's right. Discovery was a while ago, but this new kind of like actually the richness of the diversity of archaea has really come into better focus because of these more recent, kind of in the last 10 years, I would say deep sea expeditions and what
Mm,
robotic sort of sampling. What's wild is the ability. I think there's a lab in Japan and a few other places where they take, they not only are they sampling or the challenge, right, in thousands of feet of water, is to then recover these things and keep them alive. And so that was one of
yeah,
the sort of pivotal advances that we are diving into on TWiEVO 91 was, to grow these cells. In some cases, I think they take like a year or months to divide from one cell to two to four.
yeah, right.
Um, but then, sort of getting them just to sort of make it for long enough to then collect their DNA, their nucleic acids, and start to ask, okay, given the genomic analysis, how does this fit into that big picture? And so that's when, so what we were talking about there was sort of in the first pass at some of this analysis. It really scrambled where eukaryotes, our cells, sit in the kind of big picture of supergroups or kingdoms of life. And so it put the eukaryotes were actually nested within that category of archaea. It gets to your point about them seeming more
Mm,
eukaryotic like. That's probably like reverse the flow. The eukaryotes sort of emerged from potentially these cells. And then the relationship that then with bacteria becomes a little more sort of adjacent, was sort of the gist of some of these studies based on those discoveries. But so what that, the seed of advancing the field here was getting those sequences for the first time that sort of fills in those branches on the tree of life. So kind of new discoveries of life around us. I think that's still ongoing. There's probably all kinds of, no question there's missing branches, but there's probably missing, like how big, like near the trunk, so to speak, where there are like categories of life that we're still,
yeah.
haven't really glimpsed at. Yeah.
Let
Yeah.
me put up this image from the web. It's a
Uh-huh.
phylogenetic tree of all these. So we have the eukaryotes, and then the archaea. As you can see, they're on the eukaryotic branch there. Can you see that, Nels? Yeah.
Yeah, I can. Yeah. And this is great too. So at the base of the tree you're showing, we're going to use a slightly different alphabet soup, LECA, or this is LUCA, the last universal common ancestor.
Right.
LECA, which we'll be discussing today, the last eukaryotic common ancestor. So that comes up
Yep.
a little bit. So I think this tree that you found is maybe a little bit earlier. So the difference,
Yeah.
so take that brown branch, the eukarya,
Yeah.
and drop that in somewhere to the red category. That's what I'm trying to say about, or what I mean when I say it's nested.
Oh,
In,
so it's
yeah,
branched off of the red. It's not a. It doesn't have a common
indeed.
ancestor, but it's actually branched off of the archaea. Right.
That's right,
Got
exactly.
it.
So, and these still are not, these are putting together these trees is tricky at high confidence, and so think of each tree as almost a
Mm,
hypothesis. When we draw these things, and it's an idea that could be revised. It's sort of, we hope that tree represents something like that's closer to the truth of how life sort of started with common ancestors and then diverged. But that's constantly getting revised as people are bringing new sort of techniques or ideas or new analysis, new data into the conversation. And so that's, yeah, I think we were looking there at sort of pre-Asgard archaea. When you put those into the mix, it sort of shifts the eukaryotes more kind of embedded in that
yeah.
red clay.
That
Yeah,
that tree I just showed you was proposed by Carl Woese in the 90s.
there you go. Yeah,
He had discovered
yeah.
archaea. He was he was interested in microbes that make methane, methanogens, right?
Yeah.
And he was sequencing the ribosomal
Hmm.
RNA,
Hmm.
and which was very hard to do back then. And he got the sequence, and he said, these things aren't even bacteria. The ribosomal RNA is totally different, and that's where
Yeah.
he realized these methanogens are not bacteria. And he called them archaea. And he got a lot of pushback over the years.
I know it.
Amazing.
Yeah. No, and it's really remarkable given, like, as you're pointing out, the difficulty in getting that data, right? And then in the limited sort of scope of that data, but then to actually what Woes and others were able to sort of put together pretty incredible rough draft of what we think about how life
Yeah.
is organized today. It's really impressive. And now, that's we're kind of in this era where I would say, so in addition to all of the new discoveries of these critters like the Asgard, these other Norse, just named for these Norse figures in mythology, um, there's also, our computing power has come up a few notches since the early days as well, since the 90s, as we're 3 decades on. And so that's sort of the other major category, in addition to sort of adding new branches, is then to be able to analyze as you're really filling out the tree or sampling more, or more rich sampling of the tree, having the computing power to crunch through it. And then maybe, a third sort of area here is also just becoming more sophisticated in the analysis. So what are the sort of, ways of making comparisons that have some statistical rigor, or,
Mm,
and sort of account for what's necessarily an incomplete picture, and also, a picture that's like sort of throwing fog or sand into what really happened. We have no time machine here, and genetics is a great tool, but it's not perfect. There's all kinds of rule breaking in vertical inheritance. The most obvious is horizontal transfer, but there's also these symbiotic events, genomes colliding, and then genes moving. So a lot of the genes, right, encoded for mitochondrial functions are no longer in the mitochondrial genome. They're in our genomes, the nuclear genome, and so that also hints at kind of the scrambling of the signal in a lot of ways here. So growing collections of genomes, refining comparative approaches, and that's this preprint. That's where these authors pick up the story. And so they describe here what's kind of called in the field of phylogenomics approach, where you're trying to not just think about sort of one gene at a time, but multiple of them. And I think sort of break, kind of the getting to the punchline a little bit by the analysis we'll walk through in a bit of detail. Adding, I think the real advance here is adding a lot of sequences from viruses and trying to see how that fits into the sort of the proteomes that are shared in the modern samplings today, which implies that they trace all the way back to a common ancestor. And the modern viruses of today encode some of these genes, and in particular, the DNA viruses that have large genomes. And so these are some of my favorites. I think some of your favorites too, Vincent, as we've crossed paths at the giant virus meeting
mm,
over the years. And in fact, that's sort of how this preprint came onto my radar. So Eugene Koonin, who's been on TWiEVO a number of times talking about some topics related to this. He posted this on Facebook. This is sort of the random walk of TWiEVO. How do we pick our papers? We love hearing from folks who are showing up on the live streams or writing us emails. But we're also kind of just comparing notes or social media feeds with our kind of science heroes and colleagues, et cetera. So we'll dig a little bit deeper as we get a little bit farther into the viral contributions here. But the data, that the analysis that the authors do here also kind of paint a more complex story from where we kind of left off in the TWiEVO discussion last time about the sort of simple idea that there was a archaeal-like critter, an ancient critter, that engulfed an alpha proteobacteria, which became the mitochondria. And I think in some ways, it's a field that sort of goes between simple and complicated, or, really imaginative and more data focused. The Asgard archaea might have been a moment of sort of simplifying based on those new things. And I think now, as this group will make a case that there's
Hmm.
more going on under the hood here based on the analysis they do. Okay, so let's see how to go from here. So the the real or to kind of break down the approach. So it gets complicated in a hurry, and my God, the amount of sequence data that they sort of start with at the open part of the funnel, and then sort of try to distill that into comparisons that add to this. So they take to do this, they take a approach where they try to define the proteome. So the proteome, we're now at the protein level. Protein sequences are really useful for comparative approaches, phylogenetic approaches. You cut back some of the noise of, and especially when you're thinking over long time scales, the codons, the DNA, you can make a lot of mutations that, because of the degeneracy of the genetic code, are actually the same amino acids. So you have sort of 4 characters with a little more, sort of wiggle room for mutations and what that means for evolution. The amino acid sequence gives you 20 plus characters, 21 characters, if I'm remembering my amino acid numbers, and sort of in some ways more robust comparisons to try to make the case that these proteomes are orthologs. And so that's sort of the first pass through here, is the authors go sort of protein family by protein family, and then try to define as many proteomes, so all the collection of proteins that were shared among modern species. And that's, that number becomes pretty arbitrary, or not completely arbitrary, but it's dependent on your
Hmm.
sampling. And so they build here.
So this is called,
Um, yeah.
first of all, it's LECA, the last eukaryotic, right?
Yeah.
It's
So
the
you
ancestor. So
correct.
why
Yep.
did they change it to eukaryotic and not universal? They want to be
Well, so
specific in this case.
yeah, so universal, that's trying to get to the origins of life, right?
Mm,
So the universal would be what were the first cells like
right.
that then gave rise to bacteria, archaea, eukaryotes. Here we're moving one node over, and we're
Okay.
saying at the base of the eukaryotic tree, that's so universal becomes eukaryotic. What was the first eukaryotic cell? And then
Hmm.
now, as we sample, so this like ranges from our cells, multicellular critters, all the way down to single cells that are eukaryotes, things like the ciliate. So there's like paramecium and a lot of pathogenic microbes are eukaryotes as well, single cells,
Right.
things like plasmodium that causes malaria, etc. So yeah, so from LUCA, LUCA to LECA is a jump
Okay.
one node on the tree. Yeah.
And then they
So
say
yeah,
they
yeah, yeah,
they infer the LECA proteome, right? And you just kind of said that.
yeah.
Basically, you take bacteria and archaea and eukaryotes that are around today, and you take, I guess, the most common proteins, and you say these must have been present in LECA, right? Is that the idea?
That's the idea. Is so, yeah, exactly. It's so so the way they put it is they reinfer the protein families that were present, and so that would mean that you find that a member of that family in each of the modern eukaryotes that you sample. Might be missing in bacteria because that would get to LECA. It might be missing in archaea, but it's in all of the eukaryotes. And I think there's
Okay.
something like 7 supergroups. The classifications get pretty complicated in a hurry. And just to like step back for a minute, to give a sense or to put in numbers the amount of data. So they're refining the analysis using the like 65,000 pangenomes of bacteria. So pangenomes, that's what's the pan part. So bacteria are complicated critters in a lot of respects too. So if you sequence one genome, you might find something that you don't find in another
Mm.
bacteria of the same, like E. coli, right? The same quote unquote species. And so the notion of pangenomes is as you keep sequencing, you keep adding genes that encode proteins, part of the proteome. And it's that collection that you might not find in any single one member of the species or population, but in sort of the collection of them becomes the pangenome. So 65,000 of those. And you might say, okay, well, wait a minute, bacteria aren't eukaryotes, but they actually want it for this analysis. They want to go back closer to the LUCA because it's the bacteria, the archaea that are contributing. The other primitive cells, right, are contributing to what then becomes or the origination of the eukaryotes. But there'll be a lot of families missing from the branches before, but they also share obviously a lot of these, and so that's where the analysis is, and
But
they have some yeah.
if the bacteria just contributed mitochondria, then nuclear coding genes shouldn't matter, right?
Yeah, so that's one of the we're getting to the kind of punchline or the take home is it's not just and people have said this before. This isn't totally new, but maybe what's a little bit new here is the amount of contributions from other major families of bacteria might have been underestimated. And so it's now that you have these 65,000 pangenomes, and then the sort of analysis that they propose is, whether it's certainly not going to be the final word, but the kind of comparisons they draw, they think are showing the signal in a way that hasn't really been seen
So that
for the
they're
first
not
time.
there are other genes besides mitochondrial genes that have been inherited. That's the idea.
Exactly. And these could be other symbiotic events that are now,
Hmm.
that are sort of reversible in a sense. This can also be horizontal transfer, meaning that all of these, and actually we'll get to sort of almost the landscape or the environment that they propose where LECA was emerging, the last eukaryotic common ancestor. Um, and so, in addition, and so and getting at your point where it's not just the mitochondrial genes, how do these things get here? What's the sort of mode of transport? And so here, what they're proposing are really kind of fundamental in the experimental design is to include the virus sequences in the analysis kind of straight away from the beginning. And
Hmm.
so they have 1.3 million clusters of virus sequences. I didn't do a deep dive into exactly what's in there, but again, sort of just hints at the amount of data that's going into this. So now we're talking about bacteria, viruses. There's also much less, but there's increasing amounts of archaeal sequence, and then also all of the major families of the eukaryotes. And here it's almost one of the challenges is not including everything, especially as it relates to the eukaryotes, where we have probably the most sequences, or well, depending on how you slice it. But to sort of sample like a balanced sample where you're including representatives from all of the modern eukaryotic groups, and how you include these or don't include these have like a really big impact on the number of protein families that you sort of infer, and then how many of them you include in the comparative analysis that comes next. And so the way they did this is they I think truly wild how much data they curated here. And they end up with 3 data sets, with 300 proteome data sets, and they're about 50% overlapping. So they're not completely independent data sets. And so they're including, some of the sequences that we just were discussing, but also proteins from at least 5 species of eukaryotes to aim for this balanced sampling. And they want balance, meaning roughly equal relatedness between the modern groups, right? And so the motivation for having kind of 3 overlapping is to kind of take, as they do their analysis, build trees basically, and try to figure out the relatedness of these things, who contributed these based on how it's related to the modern eukaryotic proteins. They kind of want to come at it slightly different angles to try to see how much signal there is. So if there's something shared in these overlapping data sets, they might think that that's more robust because it sort of shows up in all 3 cases versus the necessary noise that you're going to see because there's just tons of sequences here. There's tons of time as you try to stretch your analysis back retrospectively this far.
So speaking of time, Nels, how long ago did LECA exist,
Whew, yeah, I
roughly?
meant to look. Yeah, good question. I meant to look that up on my pre-show sort of maneuvering here. I think it's roughly a billion years, but I'd be happy to be corrected. I think that roughly a billion years sort of slips up.
2
2 billion.
billion.
Okay, let's double that. Yeah, adding a billion years, like 1 + 1 is a ton more time. Yeah, so
Yeah,
we're
1 or 2 billion. What's the difference,
let's
right?
among friends, right? Yeah, yeah. So, and I think the current estimates for origins of life, LUCA, is more like 3.5 to 4 billion years. Again, these are sort of moving goalposts, which even if you move a million years, that's like, it's just sort of boggles the mind. So, okay. So yeah, if accounting for 2 billion years, there's necessarily tons of noise that you're up against here.
So let me
Another,
just uh,
yeah, please.
clarify. So, for example, one of these proteins would be a DNA polymerase, which all of these organisms would have. right?
Yeah,
And
absolutely.
so you can take DNA polymerases from contemporary bacteria, archaea, and eukaryotes, and assume that they were in LECA, right?
Yeah, and viruses, by the way, right? They have DNA
Yeah. So
polymerase
yeah.
too. And so all of these shared proteins that show up in these, like all of the diversity of modern life, how are they related to each other? And I think, to try to distill it, if that DNA polymerase that we see in a modern eukaryotic group looks exactly like one that you see in a virus, um, the notion is not necessarily that that exact sort of family of viruses was there, but that there's transfer, that
Mm,
these viruses are sort of gaining and losing this as part of the original sort of traffic patterns in a sense. And then this also gets at the bacterial genes, for example. If they're all from alpha proteobacteria, that would be closer to what you were kind of putting up there with, oh yeah, it's the mitochondrial event. So this ancient cell engulfs what becomes the mitochondria, and most of the genetics is there. But what they're seeing is at least three other groups that have like kind of big contributions. And so that's what we'll kind of unpack here. It's important. I think it was really important, the point you made, Vincent, about putting some functions on this. What are the
mm,
sort of shared things? So in addition to DNA polymerase, this is what's copying the DNA. RNA polymerase is sort of these fundamental core functions. There's a lot of metabolic core
hmm.
functions. How do you get energy from the
Yeah.
environment? And so they spend some time, or a significant amount of effort, to not just sort of say, okay, here are the sequences, but here's what they do. So they're trying to curate some of the functions out there. And so they build the trees based just on the sequences, but then they very quickly move to the acronym here is OGs, which is orthologous groups, right? So here, what they're trying to do, different parlance and most modern, not outside of science, but
Yeah.
the OGs, the orthologous groups, that's a prediction that the same gene was in LECA, at the origins, or the very early days of eukaryotes. And then that gene diverged as all of these species diverged going forward. But it has that sort of, has that heirloom quality to it, that it was there at the beginning. And then we see just the appearance of that performing these core cellular functions with some variation as as species have diverged. But the ones where it looks more closely, or how these relationships match, might give some clues about where these came from before LECA emerged, as all of the sort of stuff was coming together. So that's sort of the first step, is to characterize it that way. But then to finally get to the function, they move to what they're calling KOs, or KEG orthologs. And so this is now nested alphabet soup. It's an acronym within an acronym. The K, what's a KEG ortholog? KEGG. That's short for the Kyoto Encyclopedia of Genes and Genomes. So they just say KOs instead of Kyoto Encyclopedia of Genes and Genomes orthologs. But the reason they do this is because that encyclopedia is trying to get at the functions. So
Mm,
of these orthologs, these shared genes shared in modern species, what do they do? And so that's where the KEG identification will say, oh yeah, that's a DNA polymerase or a subunit of it, or that's involved in this pathway, this part of metabolism. And so what's shared in what's there says a lot, right? What today could say a lot about what was there at the beginning. Some of the dangers, sort of the obvious sort of danger, is that it's missing in some groups and present in others, not because it wasn't there at the very beginning, but it's been lost in some of those groups today.
mm.
And so you might say, oh, well, here's a whole branch of eukaryotes that don't have this sort of metabolic pathway, so it must not have been there. But they're given the way they're analyzing the data, what they say is, oh yeah, no, it was there. But then the sort of easiest explanation based on the data in hand is that in this specific group more recently, they let go of that. Maybe there's another set of genes or proteins that are accommodating that function in some of these lineages. And so that
But
also kind of highlights the challenge.
but they're assembling this LECA proteome based on contemporary proteomes. So then you have a polymerase from a archaea, eukaryote, and prokaryote. Which one do you put in the LECA? of the three.
Well, so they don't do this, but what you can do is, depending on the data you have, you can try to reconstruct an ancestral sequence,
Okay.
um, right? So
Yeah.
you
Right.
basically ask, um, you know, working backwards using statistical arguments, which can break
Mm,
down, especially over long distances of time and large divergences between some of these protein families, even the conserved ones. But you say, we think that in that doing a reconstruction, it's
okay.
most likely by a probability that this amino acid was there. Yeah, you can do that level of analysis. Here they're really using the comparisons to try those relationships and kind of messing with them to try to
Mm,
get a sense of what was there. And so just to, put some numbers again on like how big are these proteomes? So how many proteins are they inferring were there? It really depends on how on your sample. So they say that in their inferred proteomes, they could get up to 9,400 plus proteins if they just use 3 supergroups of eukaryotes. And then
mm-hmm.
that number drops to about 5,500 or so proteins if they include 5 supergroups. And so that kind of gives an example of like the more you include, the harder it is to sustain all of the shared ones because there are these losses along the way. And so that's really, I think that's kind of the game here, or the advance maybe, is that they're trying to like add and subtract. So first of all, define protein families again on the expanded data sets that are available, and kind of growing by the day or growing by the month. You could do this again in 5 years and probably get some pretty major revisions on some of the relationships of what's being proposed. But then by kind of adding and subtracting, running the trees or comparisons again, and trying to use what sort of kind of persists in these cases to sort of prioritize what's the signal from the phylogenetic noise or the comparative noise that's out there. And so in the end, they kind of settle for kind of the knock-on analysis on about 7,500 KOs or KEG orthologs, functional groups, to then really do sort of the heavy lifting of the comparative analysis in the paper. Okay, so finally, Bob, there's a lot of words to just get to figure one, but that's okay.
Boy, the figure one is really something. Let me show you.
Whew, yeah, so kind of a tangle here. It's
Oh
actually
gosh.
kind of it's really interesting, Vincent. So in this moment, for most of the papers that I've come across in the field, you don't see like this looks like a subway map, right? Like a really like the subway
Gosh.
of like
It
a
is
really
amazing.
complicated city. This isn't a tree. This is trying to capture what is the metabolism. So we can read the um,
Mm,
the figure legend reconstruction of the ancestral proteome of LECA and its metabolic features. So this is close. This is like kind of what biochemists would do um, in a situation like this. It's sort of meant to try to capture what was going on in these ancient cells.
it looks like a
Yeah,
circuit map, Nels.
yeah, exactly. And so if you really dig in, these are the enzymatic pathways that
Hmm.
are involved in taking resources from the environment, atoms, molecules, and converting them to energy, converting them to ATP or to other sort of metabolites that the cell can use for energy or use for communicate, basically sort of taking the energy around you and putting it to use as a living
Mm.
function. And so, yeah, and the color codes are sort of the different major categories of enzymatic classes or pathways that produce metabolites, things like ATP or like you could we all probably have sort of not all but many of us horror stories of when we were in intro to biochemistry and tried to make sense of these like subway maps. But what this is really wild because so there's if as you zoom in and purple is central carbohydrate metabolism. There's how do you deal with lipids, right? Processing the
Yeah.
fats. You've got, of course, glycan metabolites, so dealing with sugars and how these get converted to energy.
I think that
It's
circle
also,
is the Krebs cycle, Nels.
that could be. I wouldn't. That seems like a familiar sort of feature of these kind of subway maps. That's right.
Mm.
And then even behind it, right, and so in gray, kind of grayed out, are other modern um, parts of cellular metabolism that aren't shared, right? And so what they're doing here in the color coding is again trying to making that distinction of here's what was there potentially at the beginning. And so not necessarily because we find that in all modern species, but because you see it or in enough of them,
Mm.
and in order to say or to make the case that in other cases, the yellow part of the map is part of amino acid metabolism. It won't all be lost, but there might be some of the subway stations or some of the track between subway stations are missing from some, but they're inferring that it was there for it to make sense for
So
sort
so
of.
basically, this is the reconstructed um, proteome of LECA, right? This is all the
Ex-
different things that they think would be encoded in proteins.
exactly. So kind of hiding in this behind this map are 7,500 proteins that
Hmm.
encode the enzymes that are modifying lipids or processing sugars and so forth. A lot of nucleic acid chemistry is going on here, which then gets incorporated. Not just for sort of energy to do reactions, but information.
Mm.
As we put together RNA and DNA and use that as the genetic code to replicate and then move these cells on to the next generation.
So Andrew wants to know, what are the lipid metabolism? What's going on there with those lines going up and
Like the radiators, yeah.
down?
That's a great question for a biochemist who works on lipid metabolism.
So
I'm not
Barb
going to.
Max says, I think
Yeah,
that's where lipids have fatty acid groups added step by step.
yeah, yeah.
Yeah,
Yeah.
that's
And
probably it. Yeah.
it's almost kind of represented in that pattern that you see there as you're, and as like cholesterol, for example, right? You're adding to the chain and it's becoming saturated or unsaturated. That's trying to visualize those pathways that
So
make.
so these are keg groups, right? Uh,
Correct. That
so
encyclopedia.
how do you
Yeah.
use this to then say, where do they come from, right? Which is what they want to know, right?
Well, that's what's really interesting here. So, usually in a paper like this, what you see isn't a subway map, but you see a tree. And that's what's kind of noticeably absent from the main figures or any trees. And I think that, that's a deliberate choice by the author. It's really an interesting take on how they're sort of framing their study is to step away. So they're doing the trees, but there's thousands of them, or
Yeah.
or at least depending on how you slice and dice it, three of them with the 100 proteome data sets. And I think they're trying, and those trees will have a lot of complexity and incongruences, et cetera. I think they're trying to sort of step away from that and bring kind of front load the KEGG pathway, some of the function,
Mm.
and to put it in sort of into the category of metabolism. And one of the potential, I think, advantages of doing that is that now you're starting to think about, well, what does a cell need, to live, basically? And you're adding some of the functional sort of, parts of the equation
Hmm.
right up front. And then that's, we won't, let's not go any deeper into figure one, but then they start to then say, okay, for this pathway, then how many are contributed by, this group of bacteria? How many are contributed by this group of archaea? How many are they found in the virus sequences? And not just found there, but are like the most closely related. And so then what that allows them to do, I think, is to build an argument that, based on all of the metabolism you need, that it's not all coming from one place. It's coming from sort of multiple players as they're inferring this back. And so what that suggests then is that there's, additional gene exchanges. It's not just that one engulfment of what became the mitochondria. There's genes coming by horizontal transfer or other sort of associations. The viruses are acting as kind of genetic diplomats, shuttling information back between the ancient bacterial-like cells, the ancient archaeal-like cells, the beginning of the eukaryotic cells. And so kind of hidden in that subway map, and the yellow color is sort of a mosaic or a different, like a quilt of different genetic contributions. And because they framed it that way, excuse me, it allows them to sort of go at this sort of in a different direction. Okay, so we're at, they've sort of capturing and summarizing all of the functions through the lens of metabolism. And so what are some of the take homes, I guess? And, I would say there's not a ton here, honestly, but there is one important one that they kind of lift up. And that is that they are suggesting that LECA, that last common ancestor, was aerobic. It was, so as you put together the metabolic picture, it needs oxygen for this metabolism to work. oxygen is part of the chemistry, right? And what's missing is some of the anaerobic pathways, which have, I think, for decades been sort of assumed to be part of the um, story of the eukaryotic cells emerging, is that they might include that based on probably just subsets of things that have that, and the notion that these critters kind of feel primitive or feel ancestral, and so that must have been around. um, and so yeah, so that was one of the, I would say, take homes, is that they are not getting a lot of signal for these genes that are able to work in anaerobic environments, and so that might say something new about where the LECA was emerging, sort of, or what kind of environments it was in. They also compare. So the other sort of major comparison that I think is important to highlight here is the modern free-living single cell groups. So LECA itself is a single cell, not like our cells that are part of these conglomerations of trillions of billions of cells or whatever it is. And so there are three main groups. Actually, I'm just behind on nomenclature or how people categorize these. Have you ever heard of these groups? The fluas, the fluos, and the floops?
I have not.
Yeah, me either.
Mhm.
It's kind of catchy, but so the fluas, that's short or a new acronym for autotrophs. And so these are cells that can produce energy. So plants are obvious or multicellular embodiments of this, but single cells that do photosynthesis would be in the category of fluas. The floops are phagotrophs. So these are cells that eukaryotic cells that engulf other cells. These are some of my favorites, the ciliates. So things like paramecium, these pond critters that swim around and gobble up the bacteria and viruses and particles that are in their environments. And the fluos, anyway, these are osmotrophs. So these are critters that secrete enzymes and then absorb nutrients by osmosis. So it's a more kind of subtle way to gain energy. So either you make energy, you sort of swallow it in big gulps, or you sort of bring it in by osmosis through like small molecules crossing the membrane. And this includes a lot of bacteria, some fungi, and algae as well are these fluos. And so that's our sort of these three major groups, and so modern groups. And so the question is, okay, well, what did LECA look like relative to those? When you at the proteome level, the collection of proteins encoded by the genome. And sort of the take home here is that for the fluas, the autotrophs, and the floops, the phagotropes, the energy producers, and the gulpers, they have expanded proteomes. So they've sort of and when we think about some of this complexity too, right? They've added these genes that encode these other parts of metabolism into their pathways. And they've had a lot of time. They've had 2 billion years to come up with some of these things, and selection acting on this as sort of success stories for more metabolism. In contrast, the fluos, the osmotrophs that are doing osmosis, they appear to have lost a lot of genes relative to LECA. So they've let go of some of that complexity that was already there in that primitive, in that sort of ancient ancestor, but still retained some of the sort of core pathways that are going on. It's kind of just interesting how the, sort of once you sort of define or infer what the proteins that were there kind of at the beginning, there's, how things have been either added or let go of. In terms of additions, they also point out that all major three groups now do a lot more signal transduction. So they're the genes that are, allow you to take cues from the environment, signals, transduce them into responses, cellular responses. So you might, if you're in an environment with a ton of oxygen, the increased oxygen level might be the signal. You might transduce that signal into upregulating hundreds of genes that then, are able to take advantage of some of the aerobic respiration to make ATP, things like that. Okay, so taking stock at this point, is their proteome better than previous work? Probably. They make this is outside of my wheelhouse. I want to confess, like, I'm sort of a hobbyist who's sort of surfing in and trying to make heads and tails of it. So I'm guessing, in some ways, yes, that their proteome sort of captures, especially I was really intrigued by the addition of all of the viral sequences. And I think bringing that more kind of central, like taking the risk of centralizing that in the genetic conversation, so to speak. In some ways, they're probably, have missed the boat. This is, again, the scientific process, right? Where the truth lies out there 2 billion years ago, plus or minus. Us trying to see that today
Mm.
has massive challenges. And so, probably fits and starts. In some ways, we're kind of getting closer to what might have been. In other ways, we're kind of deceiving ourselves because there's all kinds of twists and turns in the story of life, the story of eukaryotes along the way.
So
Yeah.
Nels, I like this figure 2 that kind of summarizes the
Yeah.
origins, right? On the left here,
Agree.
panel A.
Yeah, this is really cool. So now they're using their data sets to take a kind of deeper dive into the ancestry of LECA. So where are the contributions coming from? These are like the contributions right before the origins, and then that sort of have then mark what was shared. So what they like to break down. So this is in panel A. So what they say is that 40% of the proteins in their proteome are only found in eukaryotes. So that's actually the blue box, and panel A there, there's that big green um, rectangle. And those are all the contributions compared to the light blue, which are the innovation
Mm.
so the genes that they, or the proteins, I should say, this is proteomes, proteins that they only find in eukaryotes. And so that those are basically, because they're missing from all of the other, from the viruses, from the archaea, from the bacteria. The idea is that this is sort of evolution forward in the after the eukaryotes branch off
That's what they
from
call
everyone
innovation,
else.
right?
Exactly, they're eukaryotic innovations. They're specific to to eukaryotic cells, and that makes a lot of sense because now you've
Yeah.
sort of totally rewired all of your metabolism. You have like the proto mitochondria, and everything is organized. The membranes are all coming. That lipid metabolism is all sort of coming online in new ways and being selected on. And so those are those 40%. The other, so these are another or way of characterizing it. They call them ESPs. The acronyms in this field, it's just like over the top. But so eukaryotic signature proteins, almost 1,200 of those. So it's again sort of like almost nested variation coming out from these advances. So that's like a little under half, the so-called what they're calling the innovation class. And then to get to 100%, you have 60% in the acquisition class. So that's the green part that then you can break down. And sort of that, so 100% of that 60% is 40% bacteria. And of course, the alpha proteobacteria, what was sort of historically thought of as the mitochondria, is a big contribution there. 10% archaea, and 10% viruses.
Hmm.
And so that's like that. Those numbers are proposing that sort of dark blue box is the same as that. I don't know, is it chartreuse sort of dark yellow box is from viruses. That's
Mm.
I think that's a pretty wild idea, actually, that it would be that big. So certainly, I would say up until now, I don't follow it that closely, but people would concede that viruses were in this conversation in the origins of eukaryotes, but that it would be an equal sort of contribution as the archaea. I think that's pretty wild that their analysis, because stepping back, I think you have to say, well, they included a ton of virus sequences for the first time in that analysis. And so of course they see more virus ones. And so then the question becomes not are they going to be there, but is it real? Is that real signal? And that's probably, where the next sort of set of arguments, or as peer review is, I'm sure, unfolding for this preprint now, that might be where part of the criticism will be litigated. Is that really did they do their comparisons in a way that they're not just adding a lot of these sequences and saying, oh yeah, they must be part of it, but that there's real evidence for that. And that's what comes out of that deeper kind of analysis that they do.
So the fact that, say, bacteria contribute 37%, yet they can only account for 1, 2, 3, 4, 5, 15 or so percent, means that they're missing some, right?
Oh yeah, absolutely. And that's, maybe not missing, but they can't trace it to who contributed that,
Yeah,
because there's
right.
just right. It's about this equal relatedness between those other groups. And so I think the way they kind of tackle this, right, is to actually say, okay, you see some of the alpha proteobacteria, which is, I think,
Yeah,
so now we're doing the third set of bars over, is the deep red one there. And that's like, feels pretty familiar. That's been implicated in sort of being became the mitochondria, this like core and symbiotic event. But then you have, that's what's new here, is you have at least three other main groups, or even, I
right,
guess, illustrated here,
right.
four, that are basically, again, just like those viruses, are like kind of at the same weight class in a sense as the archaea. Here, the other bacterial groups are in the same weight class as alpha proteobacteria.
Yeah,
And so I don't think they're not saying that, that sort of dispels the idea that it was a ancestor of alpha proteobacteria that became mitochondria. But there's more going on here under the surface. Were there, other temporary engulfments, or all kinds of, collisions? So horizontal transfer that then turned into vertical inheritance, like that sort of, I think it opens the door to a lot more
hmm
genetic traffic, basically, and points to the viruses, like, again, as potentially a source of that, which is what I think that just
cool.
sort of kind of sparks my imagination and kind of gets
Yeah,
me interested
cool.
in it as well, right? And so here's where it gets, for me, I think, even better, right? So of that viral contribution, half of it, or like a little over half of it, is from a single class of viruses, the DNA viruses with large genomes. And so what are these? These are the giant viruses, um, viruses that can have, encode 1,000 genes in their genome, in some cases more than free living bacteria. Um, the reason these haven't been in the conversation is we didn't know about them until relatively recently, right? So as Carl Woese and others were doing some of this original analysis, the giant viruses weren't there in the
Yep,
conversation just
yep.
because we hadn't discovered them yet. And so this is, again, how, the discovery of new genetic entities or critters, new cell types, et cetera, really can kind of change your perspective. What's beautiful about these giant virus genomes, and you've talked about this a lot on TWiEVO, talked about it here a bunch on TWiEVO as well, is that when you do a catalog of those, genes encoding, say, 1,000 proteins, they're from back. You can link them to bacteria, you can link them to
Right,
archaea,
right.
you can link them to eukaryotes, and so it's sort of this conglomeration, right, that sort of hints at all of these collisions and the exchange of genetic information. It sort of breaks the rules of mother to offspring inheritance. And so the
Yep.
fact, right, that it's this class of viruses that are sort of like gene hunters that are coming up, as like, that kind of supports that there's some signal here, that it's not, all of the small RNA streamlined
Yeah.
genomes,
Yep.
et cetera. And
Yep.
so pretty interesting. Okay, so what? Finally, I won't. We're already past, or more than an hour in, and won't belabor it. But the next real question, right, is what's the robustness here? And so of the data, that, so they try to, how they describe it is like a stress test for the data set. Um, and so they try to take away, you know, so for the groups of genes that are at least 15 plus, the functional categories, like, so if it's lipid metabolism, if it's to sort of mess with the lipid tails, does that involve 15 or more enzymes? They take some of those groups away, off the table, and then rerun the analysis. So they describe this as kind of negative controls. It almost felt like a variation on bootstrapping. So in simple phylogenetics, where you're just taking, for example, one gene, um, and when you see those trees, scientists are drawing these trees, and it has these numbers at each of the nodes. That's called a bootstrap value. And so basically what you're doing is you're removing some of the sequence from that gene and then asking if the tree has that same topology. Does it have the same arrangement? And every time the answer is yes, you add another number to that. And so you might do it 100 times. And if it's 95 times, you've taken out different segments of the genetic sequence, but the topology is the same, you're pretty confident that that's the right relationship. I think what they're doing here, I'm sure I'm getting the details wrong, but it feels like it has that sort of essence. You're taking away now kind of sets of genes and then asking, do the relationships still line up, or how does it sort of scramble the deck? And so coming out of that analysis, they, actually think that there's a lot of evidence here of this vertical inheritance. So once they're sort of the pre-LECA days of a lot of genetic exchanges, but then as you sort of mark this transition into, eukaryotic cells in the subsequent 2 billion years, that these things then, there's still a lot of horizontal transfer, but there's also real signal in genes being passed from mother to offspring, sort of as you go forward as eukaryotes. Yeah. So they then move on to, sort of even a deeper dive, branch length analysis, trying to get a sense of the timing of this. I think we'll just spend a minute or two. So this is worth pointing out, because now it's like they're going from sort of, okay, who gave what, to where did this happen, right? So I think kind of, implicit into the idea that you're getting more, there's more symbiotic events. Some of them might be temporary versus the sort of what feels like the permanence of the mitochondria, the symbiotic addiction that our species like is involved in. How is this possible? If you're really gonna like exchange all this stuff, what's the environment look like where this is happening? And so they're trying to build a case, sort of in the last part of the analysis, that this was happening on microbial mats or biofilms, right? Where you've got now concentrations of species, or primitive species, that include all of these groups, and also necessarily would then include viruses that might be sort of shuttling or transporting these among this. And so they try to use the length of the branches to sort of get a sense of how this is timing out over these long genetic distances, a sense of the tempo of the horizontal transfer genes. And so in figure 3, and you can pull this up in the preprint, the longer branches are interpreted as more ancient relatedness. And so the groups that are sort of showing up there are the archaea. And that makes sense, again, for the notion that it was a sort of ancient archaeal cell that's consuming an alpha proteobacteria. They're not sort of, that dogma still stands to a large degree. And then the bacterial contributions come sort of in a sort of later tempo. And they even go as far as, I think, to just sort of hint at the idea that the timing of this sort of like mirrors what's sort of thought about, even the composition over time of these microbial mats. So it's like the right groups are there at the right time to make genetic contributions as the origins of eukaryotes is all unfolding over, a pretty long time scale, by the way. Actually, I'd be curious about what the like estimates of that are. I don't know that part of the story, or that part of the field, or if people are really tackling that idea or can tackle that in a sort of rigorous way. So maybe just to put an exclamation point on the virus, so they kind of close out then returning to the viruses. So this is sort of the microbial mats might be the place that's happening. And based on the sort of addition of those 1.3 million virus clusters of virus sequences, I pulled out a quote here that I thought was kind of fun, Vincent. So it says, together these observations depict a complex scenario involving a series of horizontal gene transfer waves from a diverse set of donors into the proto-eukaryotic lineage that was proposed by the pre-mitochondrian symbiosis pathway, right? So all of these waves of contributions, still from that's where the sort of extra groups of bacteria come from, before the mitochondria comes online as that sort of big landmark symbiotic event.
Yeah, I mean, this is not something that happens like that. It's happening over time, right?
Yeah, exactly.
I mean,
And so
you're trying different combinations, right? You get some mixtures, you see what works, what doesn't. You get more and more. It takes many years.
absolutely. And so that's the host cell that engulfed what became mitochondria might be might even be in a different group than previously thought. They go as far as to say that, based on sort of like adds more, almost like less, confidence in what is sort of the dogma in the field. But I'd say, those ideas, it gets pretty foggy. Like they're basically, almost creating fog to then sort of suggest that there's fog there, which is sort of a complicated scenario. The idea that the viruses were shuttles of these bacterial genes. So what are these genes? They also take a quick peek at that. And so they propose that there's an enrichment of kinases and proteins involved in post-translational modifications. So that's, sort of a set of functions, or you're actually changing, you're doing so to back up, you're making proteins. The proteins might be enzymes that are doing like sort of some of these core parts of metabolism. But then these proteins that are, or the genes that they're saying are enriched that have been contributed by viruses, are modifying proteins or adding phosphorylation groups or entire other peptides, what we call post-translational modifications, which gives these proteins sort of even more functions. Now, why there might be an enrichment that are sort of virus specific, again, not really clear, but that's sort of a signal they're pointing to here. So that's kind of it, and it's more, as we said. So it's historically, circling back to the start, it's a field that historically has been imagination and arguments, ideas of so not maybe at the eukaryotic junction, but certainly in the universal common ancestor, the ideas of alien contributions, meteorites landing on Earth, a lot of creative, sort of imaginative, in some cases almost like philosophical ideas about where life comes from or what it means. And then because of the lack of evidence, a lot of arguments. Now I think we're in this era of a lot of data, but where we're struggling, or the sort of cutting edge of the work, is to draw meaning from it, because especially in cases like this where you're sort of comparing to try to infer events back billion plus years, 2 billion years or more, really hard to sort of get signal from noise. And so that sort of gets to the ongoing challenges ahead. I love this for not just the biology, but the sociology, how groups of scientists, entire fields, like in the areas of origins of life, have historically just been a lot of, arguing, as I was hinting at the beginning here, and not a lot of data. And then like fields you and I came up in, Vincent, that are like, well, the data kind of speaks for itself, and what's the evidence here? And putting those two kind of almost cultures of science together is really wild. There's just a lot of fireworks, and I think a lot of potential for sparks or new insights the more we can kind of hold together or come together in new ways. And I think this paper really nicely tackles that.
This is data. These are data, right? It's a different kind of data.
Yeah.
And it's a different kind of experiment than we're used to, but putting them together is the key. And I'm not sure we're there yet, right?
I agree. So there's been, in the kind of experimental framework, like there's been some really fun advances trying to get at like, how do you sort of do like before cells? So the LECA, LUCA versus LECA, how do you set up sort of lipids and chemistry to sort of get to something that can propagate?
Yep.
And there have been some experimentalists some labs, who've really taken up that challenge. Super daring, courageous work, but with some advances. And so I don't know, like so that's kind of this home run or almost grand slam of how did life begin? And how can you go in the RNA world where you just have puddles that then are concentrating reactions, and then sort of to RNA where now you have the enzymes also have heritability or sort of propagate themselves. But so, versus the home run, which would be, is it even possible based on some of this kind of analysis, much more comparative and cataloging and sort of, comparison driven, but to use some of this to try to recreate LECA? Could you imagine sort of nudging archaeal-like cell or engineering these symbiotic events to sort of like model this, and sort of engineer, to even bioengineer this? I don't think so. See, it sounds complicated to me, but I've been really impressed at that sort of interface at the LECA stage of just, some of the chemists and the biologists and the sort of bioengineers, genetic engineers coming together and making some progress there as well.
So with this idea of viruses moving genetic material, Tom says analogy to transducing phage. Yes. So bacteriophages were the first to be shown to be able to pick up DNA from the host bacterium and bring it to another one. They call that transducing. And
exactly.
Now we know all viruses do this, including the ones we're talking about today.
Yeah, and I'm glad you brought up phages, because I haven't said it, but that's definitely part of the genetic conversation here, because these are like the shuttling for bacteria like this is, so in the sort of giant virus classes of today, there's also, phages in that conversation, certainly at the at these early sort of origin events. Yeah, it's a really good point.
So in the interest of time, I'm not going to any questions here because I think we should do some picks. But I do
Okay.
want to point out, Amy was here before. She said, "Don't forget to
Ah.
send us your video about how Microbe TV has
Ooh.
empowered your inner scientists to help with their fundraisers." So now November,
Yeah.
December, we do our fundraising for Microbe TV. Send in an audio clip telling us how we've empowered your love of science or anything that's relevant to Microbe TV. We'll play it on our programs because it will
This is
help us
great.
tell others how we impact our listeners. Right? Thanks, Amy.
Yeah. And maybe for
Thanks.
if a few of you listening right now, so we're gonna. I was just rambling way too much here, so we're unfortunately cutting the questions close. But if you're inspired to put anything in audio, tap into your inner scientist right now. This would be a great time to do it. What a great event! I love this fundraising idea.
And speaking of fundraising, Imans, thank you for your support of science communication. Really appreciate it. Okay, I think we ought to do some picks, Nels. What do you think?
Okay, I agree. Science pick of the week. So mine is just came across this. It's in beta version. It's called BioArt from NIH, the National Institutes of Health. So one of the things that we've seen crop up in the last few years are tools to be speaking of your kind of expressing your inner scientist. How do we like use graphical representations of the cells, the viruses, to put together as we're putting together our stories? How do we visualize this with cartoons? And a lot of us as scientists might say, oh, well, I don't have the time or the skill to draw all these fancy sort of combinations of things. So here, yeah, thanks for bringing this up. So here's the BioArt webpage, beta edition, but it's a collection of images that could be useful for scientists to sort of put together, all the way from, the cellular level or organelles and all the way up to the equipment that we're using, animals like that, that level, kind of veterinary, like different organizations or scales of biological organization to illustrate this and put this into our presentations. And so, yeah, so this is a new NIH public domain repository. It just has pictures that you might, you might want to draw a tick or a vial of insulin or a syringe. And so you just grab it there and drop it into your PowerPoint. So this isn't totally new. There's some private companies that have kind of come up in the last few years. And but the reason I picked this is I think it's a great resource for, practicing scientists or inner scientists to think about how we might illustrate things as we're expressing our ideas, but it's free. And so some of the, private entities that have started to do this started out with sort of a trial period. Oh, here are the viruses. Aren't these great, Vincent? Like some real style in some of these and all of the major classes. I'm seeing spike proteins, et cetera. But anyway, for these for-profit driven models, things can get complicated in a hurry. And so part of my motivation for putting this up here is just in a very small way to put in a word for the federal government being a force of good, especially, I would say, as it relates to practicing and communicating science. And so, this doesn't happen without a collection of federal employees who go out of their way to make something possible to advance science. And this is a lot of, when we think about people who have careers at NIH, who help to organize peer review, who help to kind of grease the wheels of science. In the US, we are so lucky to have dedicated, skillful PhDs, the scientists working with scientists to take the resources that we're entrusted with from taxpayers, our own taxpayer dollars, and to put that to the science that we think might lead to breakthroughs that might allow things to grow. And so I have to say, Vincent, as we're, probably not too, it's probably fairly obvious that given the election season where I'm going with this, but with, some of the noise and the misinformation and the related sort of energy that's out there, this is just one very small piece of the puzzle, one really small layer of the onion. But these folks who are working for the federal government and the NIH and the contributions that they put in to make my career possible, all of the science around us possible, I think it's all behind the scenes for the most part and has unfortunately in some circles become a bit of a punching bag. And so just want to put my small voice into the mix here to say that there's a lot of hidden value in what's going on in governmental agencies across the board. There's also, I'm not saying it's perfect or it's great or there's not, but in many ways, our system of science, which has been going for decades, more than half a century, is the envy of the world for a reason. And this is one really small example of that, the bioart thing here. But these things in the public domain, all of the genomic data for the preprint, for example, that we were talking about today, these researchers in Barcelona were able to take our understanding to a new level because of some of these efforts. And it feels like it's in a little bit of jeopardy right now, or a lot of jeopardy right now. And so I'm just holding out hope that we're able to not just sustain some of these efforts, but grow them in the years ahead. And think about the possibilities. It really hits me right now, kind of the contrast between how much sort of joy and gladness there is in this moment that we have all this technology, all these tools to see the world in these increasingly sophisticated ways, against the sorrow of what could be lost, whether that's at the environmental level or just the the ability to practice science going forward, et cetera. It's really an incredible time.
Yeah, government-funded science is not just here in the U.S. It's in most countries where you do science. That's a great way to do it, because when you privatize, then you have all kinds of issues, and that is one of the issues in this upcoming election. One of the parties believes in extensive, less government and more privatization, which of course enriches your pockets, but that's not always the best way forward. At least for science, it's done really well being a government run, not just here in other countries. Yeah, you can find ways to improve it, but you don't want to change it to be nonprofit.
Yeah, and it's wild. I think it's so, like, the things that you might build on over decades turns out is in some cases relatively easy to get rid of, but much harder
Oh
to
yeah.
build. All
Oh
right.
yeah.
And that mismatch, this is not a symmetry. And so, potentially letting go of some of these things in the face of efficiency, or this is how we do it in industry. There's some really good reasons to have things in the public domain or public, sort of enterprises where private sort of impulses don't really scratch that itch of what can be
Yeah.
possible, of what we can
Yeah.
build if we sort of open our minds a bit. Yeah. Anyway, that's my science pick of the week, and wishing everyone, the best in the weeks ahead. That our country feels like every year or every 4 years we find ourselves almost staring over this cliff. But just hoping everyone is able to be safe, to step through all of this, and that we get a good outcome on the other side, and can continue to build in ways that are improve on things that certainly can be improved. How about you, Vincent? Speaking of building, what's your science pick of the week?
My pick is an article in Science Line called "How Beavers Built New York."
I
So
love
this is
this.
very
Yeah.
amusing, by Sarah Hashemi.
Mm.
And if you've ever taken the subway to Astor Place, on the wall are etchings of beavers. And that's a testimony to the role that beavers have played in New
Yeah.
York's history. Before European colonization, there were
Yep.
60 million beavers throughout New York State. And John Astor, the first multimillionaire in American history, made his fortune through the fur trade, including beaver pelts. And
Mm.
so all of the places named after Astor in the city come from, in part,
It's
beavers.
great.
And
Yeah.
so this article talks a little bit about the history and how it would be, right now there aren't any beavers in New York City except for Staten Island. There are
Yeah.
some, but they are beneficial, and
Yeah.
it would be useful to have some of them back again. So I think it's an amusing article. And the next time I go to Astor Place, I'll be sure to check out the beaver etchings on the
Same
walls.
here. I'm adding it to my list, putting this on my radar. This link of beavers and what's been lost in some cases extends all the way to the Mountain West. So there used to be a lot of beavers around these parts too that were hunted out. And kept out, as a lot of people, for example, if you plant fancy trees on your property, people are a little nervous that the beavers are. But so I've been involved, Vincent, actually, in an effort to build beaver dam analogs at some of the streams
Huh,
and
cool.
tributaries around here. So this is like humans on the weekend trying to kind of, get in the mind of a beaver, where we're literally like in creek beds with mud and working with county officials, et cetera, nonprofits, to put beaver dams in some of the small streams to fight against erosion, to slow down the water, to sort of recover aquifer kind of related stuff. But I have to say, as human beings, our species, were pretty shitty at being beavers. Like, our dams are not anywhere close to the level of sophistication that beavers have in sort of like, changing the ecology around it. It's like our stuff sort of just flows right through, whereas theirs are sort of airtight. And it's that right there. This is what beavers do, as have evolved to do. And so also efforts here, hopefully, and, I can imagine even, in and around New York, to return beavers to some of the habitats. As we were talking about, I think Tom brought up the case, right? Removing the dams, the human dams, in order to recover the salmon. Returning beavers, and sort of ironically to make dams in very different sort of scales and deployment, to bring back some of the ecological features that are threatened or lost. Yeah. Very cool.
All right. That's TWiEVO 107. You can find show notes at microbe.tv/TWiEVO. If you like these science programs, we'd love to have your support to continue them. You go to microbe.tv/contribute. And if you have any questions or comments, I know you like to do them here, folks, but you can also send them to TWiEVO at microbe.tv. Nels Eldy can be found at cellvolution.org and Early Bird on X. Thanks, Nels.
Thank you, Vincent. Great to be together. Thank you, everyone, for joining us. We didn't get to many questions, but next time I pledge to maybe do a little less talking and a little more question taking.
We'll be back in about a month. I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank everyone for joining us today, and our moderators, Barb Mack, Les, Tom, Vanity Nutrition. And thanks all for joining us. I really appreciate your coming and making it a calm presence here. And thanks everyone else for coming. And as I said, we'll be back in about a month. Tonight, 8 p.m. Eastern, we'll have office hours with me, where you can bring your virus questions. Music you hear on TWiEVO is by Trampled by Turtles. You can find their work at trampledbyturtles.com. You've been listening to This Week in Evolution, podcast on biology behind what makes us tick. Thanks for joining us. We'll be back next month. Until then, be curious.